Okay, so I'm going to talk about light microscopy and confocal microscopy. But just, I will give a brief overview of what we actually do here, the people who do it and what there is, and then we'll get into the microscopy itself. Then we'll have to talk about fluorescence, which adds specificity to your imaging. Confocal microscopy, which is all about getting clearer images, and then multiphoton microscopy, which is about moving into tissue where you've got a lot of scatter to deal with as well. So who are we? If you look on the internet, it's me, Sarah, and Thomas, both here in the audience. Between us three, we generally deal with most of the hardware side of things. Jerome leads up image analysis, and he works with Dina and Simeng on image analysis. So, and this is one thing you really got to think about when you're doing microscopy. It's more than just taking images. You've got to be able to extract information, quantitative information out of your data as well. So we've got a whole team that will help you get there. And oftentimes, you know, they'll have a look at stuff, and then we'll go back to the microscope and have another go at it, slightly better, hopefully. What have we got? There's a huge number of instruments. We've got about 20 different instruments in the institute that you can work on. They're all different, got strengths and weaknesses, and we can help you figure out one which is best for you. On our website, there's a whole list of stuff, so you can go and see that on the internet. So what sort of things are we actually imaging? Well, going down to all the way down to bacteria. So there's 1 micron, so you can sort of figure out how big those blobs of light are in terms of a couple of 100 nanometers. Whole cell cultures, fixed and stained. Small modern organisms. There's an image of a Drosophila embryo digesting something or other. And then all the way up to big bits of tissue, whole brain slices from the mouse brain. There's a lot of that going on here as well. And again, as I said, it's not sufficient just to get images. You've got to be able to analyze them and do some sort of quantitation of them. So here, for example, is a false color display of where software has been used to outline each cell, and then you can start asking questions about the properties of those cells. As I said before, Jerome and Dina and Simeng generally deal with that side of things. And they'll run a load of courses. So Jerome will be talking about image analysis tools on Thursday, plus throughout the year, there's regular courses on ImageJ, deconvolution, some stuff on specific microscope software, image processing in Python, and they'll also work on custom image analysis pipelines and tools. So the thing I really want to emphasize, please don't get all your data without first checking that you can analyze it. It's a bit difficult sometimes to rewind the clock on those things. All right, so down to the details. You know, when was the light microscope invented? And really, it's around 1600 that sort of usable lenses being created. So there we are, 1609. There's Galileo's telescope. In terms of biological microscopy, we've got to sort of wait till the 1560s and 1660s, 70s. For the sort of real revolution to take off. But the microscope is very simple conceptually. You know, you've got a lens, small object. This is a little bit further than F away from the lens. You get a magnified image, and that's what you're looking at. But really, the landmarks were Hooke. One, he had this is a simple lens just down there, and he looked into a world. It's really remarkable that a small bit of glass with a spherical surface gave us a window on a world that up until then had never been seen by mankind. It's really quite remarkable. All the small stuff we were not aware of prior to this sort of work here. And so Hooke, for example, coined the word cell when he looked at a piece of bark from a cork, I think that's from. This other thing. The other great person in this thing was Van Leeuwenhoek. This is his microscope. So his lens is that, his microscope is that little drop, that thing there, that's a little spherical piece of glass. And his sample was stuck on that pin. And all this contraption was just about moving the pin around in front of that lens. So he sort of held that thing up to the light and looked through it. And you saw a magnified image of it. So really at this point, biological microscopy had arrived. And for the next 350 years, we've still been looking at small stuff. We've never run out of things to do. As I alluded to just now, the magnification is just really the ratio of the image distance to the object. This is very easy to get magnifications up in the 100x. So in reverse, that's what's going on in your iPhone. This is you standing here, and the sensor in your phone is here. So it's sort of working backwards compared to a microscope. We want to make things bigger. What is special about microscopes? Well, they are designed to reveal fine details. For that, we need a couple of things. We need magnification. We've got to get it big enough so that the human eye can actually resolve the details. And it also has to have resolution. It's got to capture the fine details and relay them into the image frame. So those two things are necessary. It was easy to build magnification, but it was much harder to get resolution. And the point with microscopes is we're often trying to observe structures much smaller than the wavelength of light. So there's another special condition there. That sort of sets them apart from telescopes, for example, which look at objects a long, long way away. So these properties, particularly this one about wavelength, means there's a lot of things that have to be right in the way microscopes are made and used. So by scale, we obviously can work across lots of different scales, but we're sort of fixed tissue slices intact. Those are all things in principle you could look at with the naked eye. But as soon as we get down to single cells, this is where microscopes open the doors to a new world. And then by the time we get down to the subcellular organelles, we're looking at objects that are smaller than the wavelength of light. So down to single molecules, these are molecular machines, obviously a lot smaller than the wavelength of light. So, but there is unfortunately a sort of physical barrier. There is an optical resolution limit that we can't get past, well, until the advent of super resolution methods. But that is the limitation we have with visible light and looking at small objects. It's around about the sort of scale of subcellular organelles. So all optical instruments have got these sorts of properties: diffraction, refraction, dispersion. But diffraction is really a property, is a consequence of the wave-like nature of light. And that does set some limits on resolution. Refraction, that's what's going on in the lens surface. This is the bending of rays of light by spherical surfaces. So that's sort of Snell's law, if you like. And that leads to this whole business of optical aberrations. And then dispersion, that's the fact that different wavelengths bend different amounts depending on their wavelengths. So this is when you shine light onto a prism, you get a sort of rainbow coming out the other side. That's also a problem in microscopes, because obviously ideally you want, if you're imaging something that's both green and red, you wish the image of the green and red thing to be in the same focal plane and in the same magnification. So diffraction. What happens when you look at and the size of the object you want to look at is about the same size as the light that you're shining on at the same wavelength. This is a sort of typical diffraction type experiment. You get a plane wave, you shine it through a small aperture, in this case a square one, you get a diffraction pattern, which you can observe in the back focal plane, something like this for a small square aperture. Now, for normal optical instruments, the eyeball, microscope, binoculars, stuff like that, we're not really aware of diffraction. That's because this is going on at a scale below the resolving power of the eye in general. You don't see it on your phone camera either, because pixels of your phone are too big to capture that detail, but the microscope is designed to see fine detail, so this is a problem in the microscope. So even though microscopes have been around, say, from the mid sort of 1650s onwards, the actual knowledge of what light was up for debate for a long time. So Huygens was a sort of who championed wave-like properties. Newton was of a different opinion. And there was a lot of discussion in the sort of literature of what light really was. What was it made of, if you like? In 1800, Young did this really very, very important experiment in which what he managed to do was demonstrate that light was a wave. And once that had been resolved, all the properties of any optical instrument can effectively be inferred just from the results of this sort of experiment. And what's remarkable is that Young managed to do this just using sunlight, which is quite tricky. But anyway, so this resolved it. This told us that light was a wave. So now we had to think about how waves behave in optical instruments and how you can get them to behave a bit better so that you can get higher resolution images. Zeiss was building microscopes, in the mid 1850s. He couldn't figure it all out, so he actually engaged Abbe to help him figure out how microscopes work from a sort of point of view, a physical optics point of view. There he is. This man has made a lot of difference to our understanding of how microscopes work. So really what his contribution was to make us clear about the difference between magnification and resolution. So magnification is not sufficient property of microscopes. So his apparatus, so he had a fairly standard sort of optical instrument of the day, but he sort of abstracted the general sample you might put on there to a sort of one-dimensional rating pattern. So this was an object that had fine detail. And he looked at that object through the microscope. He put various masks and whatnot in the back of the objective, which effectively changes its aperture. Oops. And this in a sort of nutshell is what Abbe was doing. He puts a grating-like object on the stage. In the back focal plane of the lens, you see a diffraction pattern. So this white blob is the sort of zero order light, the under stuff, and then we've got this sort of rainbow due to the different colors of light in the spectrum that he was illuminating it with. And there's the first order diffraction, and then you can just see the sort of second order. As you put finer and finer detail objects on, you can see that this first order pattern has moved further out, so it's coming out at bigger angles. And at some point, if you make that finer, this thing goes outside the aperture of the microscope. At that point, no information about that grating pattern has entered your optical system, so you cannot see it. It is unresolvable. So the key was, keeping all these diffraction orders captured by the microscope in order to see the fine detail of the object that you're sticking on there. So to recast that again, here's a lens with a cone of acceptance, if you like. This is the aperture of the lens. And Abbe defined this as numerical aperture, which is n sine alpha, this angle here. He included the immersion medium, which you can sort of see gives you higher NA. So air has got an N of 1, but oil is 1.5. So the NA goes up considerably if you use immersion. Here's our sort of prototype object, just a regular array of fine detail, creating a diffraction pattern. What Abbe realized is that the resolving power of this microscope, or the resolution, is basically the wavelength divided by 2NA. So we've got this simple formula for saying what's the resolution of a microscope. Higher NA incidentally means you're capturing more light from your object, so you'll get a brighter image. So to recapitulate that all again, we've got some object on the stage. It creates a lot of diffraction orders. Fine structure leads to the diffraction. The lens only captures a limited number of these diffraction orders, so all the detail is not reproduced in the image. And this is what's known as diffraction limited imaging. So this is something we can't beat with the microscope, and it's wavelength dependent, but it's also dependent on this aperture angle here. So we want that angle to be big. Okay, refraction. Lenses bend and focus rays of light, but in real microscopes, not all of those rays end up in this perfect diffraction limited spot. So we get aberrations or distortion of that image. So how does that actually work? So conceptually, we've got some sort of point-like object, an imaging system, which hopefully adds magnification. But ideally, all those rays from that point come to a point in image space. But we can see from this a simple example that that might not be true. But just as an aside, we get very high quality pan-achromatic objectives with our microscopes. So if we use them exactly as the lens designer intended, the images would almost be free of, completely free of amylase. This would be the case, but we often don't use them quite right. As this case indicates, if we put a slab of glass in the path or the sample or something like that, you get refraction at all these interfaces, and you can start to see that those rays, rather than coming to this point here, are sort of will cross over at slightly different bits. And you can see it's angle dependent. There's much more shift of this ray for a ray that's coming through this slab at a bigger angle. So this is the source of optical aberrations, this type of effect. That's the thickness of a bit of medium of different refractive index moves rays around. And then on top of that, we're going to get chromatic aberrations as well. So different colors will move differently. You can compensate for a lot of this in the design of the lens, but it does mean you have to use the lens as it was designed. So if we think about the typical sample, we start with the objective. It's got its own refractive index for its last optical element. We've got some immersion medium, oil or air. That's got a refractive index. We've got a cover slip. We've got a sample. We've got this complex structure in terms of refractive index, which we could sort of consider as a bunch of slabs. We get things wrong. We put the wrong thickness cover glass on. Our refractive, we haven't put a mounting medium on our sample. We've got a lot of gap between the sample and cover glass, stuff like that. All of those are going to lead to aberrations. So we want to sort of make our samples as well as possible to eliminate all of these effects. So this is technique. And these are the things that we're mostly doing wrong when we're using objectives. And again, just to recapitulate that point, it's these rays that are coming in at high angles, which are the ones that carry the high resolution information that was worst affected by these errors. And then dispersion. So this is this idea that prisms, like bends different amounts going through a prism. But what that led to in microscopes was a sort of rainbow halo around objects if you're illuminating white light. That was largely fixed by this thing called the achromatic doublet, which has got a high and low refractive index glass in it. And that was cunningly contrived to actually focus red and green light to the same point. So that's a sort of color correction into between the red part and the green part of the spectrum. So around, was this 1830, Joseph Jackson Lister with William Tully basically built the first microscope that was color corrected, at least in the green and the red, using achromatic doublers. Thus far, we've sort of talked about the simple microscope, the single lens, an object, and it forms a real image up there. So this is convenient because it introduces this concept of conjugate planes. So we have an object plane and an image plane, and these are said to be conjugate to each other. More modern microscopes use the so-called infinity corrected idea. So the specimen, the object, is F away from the lens. Each point on the lens produces a parallel ray bundle. We then have a second lens built into the microscope body, which produces the final image, either up by the eyepieces or onto a camera or something like that. This bit of space here is where all the fluorescence optics go. And it's convenient because it minimizes the aberrations that those objects introduce. The objectives themselves are covered in writing. Most of them, I mean, you know, we're interested in magnification, 40x, 100x, whatever. We also can see the NA, the numerical aperture. That the bigger that number is, the better the resolution. This writing up here, Plan Apo, tells us about how well color corrected the lens is. So the more writing you have up there, the better, generally speaking. And again, immersion, it tells you what the immersion to use, is it air or oil. So don't stick oil on an air lens, that'll kill it. So the lenses themselves sort of form these sort of two general classes, either used in air, typically the lower magnification ones, or immersion, where the immersion gives you higher NA, which gives you higher resolution. So we're basically putting a bit of oil or water in this gap between the sample and the objective lens to get NA, and that's typically 40, 60, 100x. Within the air lenses, there are basically low NA ones and high NA ones. The low NA ones are very, very tolerant to how you present the specimen there. Once you get to high NA, you've got to be much more careful about how you prepare your sample. So 20x, 0.75 or 0.8 NA, 40x, 0.95. The results of these lenses is dependent on how you do it, but this one here, the 20x, 0.75 will get you down to 350 nanometer resolution easily. And this is a very, very good choice for looking at large areas of tissue. But yeah, you have to make sure you get all of those details right down here in the sample prep for the 0.95 lens to perform at its best. The water immersion ones, these are much better for biological samples because obviously the sample itself is a watery sample. So typically you're putting a bit of water either on one side on the cover glass or dipping the whole lens into the water. So here, the water gets you a higher NA, better resolution again. But then you set the correction color on the objective to match the cover slip thickness to get rid of spherical aberration. These dipping objectives are great for electrophysiology, intravital microscopy and whatnot. So in EFIS, the electrodes are sort of coming in through that gap there into the sample. We haven't really talked about the eyepiece. So the lens itself, the objective produces a real image, and then you've got this thing here, and it's just way too close to your eyes for comfort. You just cannot see it. So what the eyepiece is doing is taking that and projecting an image a comfortable distance away from your eye. So that works very simply like this. Effectively, it's just a simple lens that this gap here is less than F. In that condition, it's producing a virtual image. You can see the rays from that object diverging as they reach the eyeball. And then the eye, we just project backwards in space to an image plane, some comfortable distance away from the face, and get extra magnification in there as well, typically 10x. So that's how the eyepiece is working. So when you're looking through the microscope, if you ask yourself the question, where does the image appear to be? It appears to be somewhere in space down here in front of you. Now, one of the problems with microscopes is contrast. So cell cultures, they're basically invisible to the eye. They're completely transparent. You can sort of see that they're scattering light a bit. Thin things that you've cut them thin so you can actually see through them, again, have very low contrast. So you can use these sort of interferometry techniques to actually bring contrast to those sorts of samples. So phase contrast microscopy is typically what you're using in the cell culture lab. Differential interference contrast is another interferometry method that is used to create fine contrast of when you're in white light illumination. And it's based on the fact that most objects, whatever it is that's surrounding them, has a slightly different refractive index to the object you're looking at, or internal structure has variations of refractive index. And refractive index changes cause phase retardation, and that's the key to getting an interferometer to work. You can see that waves go in and out of phase, and you get constructive and destructive interference. So how does it work? So phase contrast, as I said, is a sort of interferometer. So I could spend hours getting bogged down in the detail here, but we're just going to go through. So the trick is to create an annular light source back down in your illumination part of the microscope in the condenser. So we have a ring of light. That goes through the microscope, the focal plane, and then light carried on through the objective up to the eyeball. Now, up in the objective, in the back focal plane of the objective, there is a phase plate, which is matched to this annular pattern. So all the light that goes through the specimen, undeviated, goes through that phase plate, and the phase plate imparts a bit of retardation on that light. But then if we think about what's going on in the specimen plane, the specimen is got a complex refractive index structure, it scatters light. So some of the light goes through the objective on paths that are not shown by this sort of annular light path, if you like. And that forms the other arm of the interferometer. So you've got the undeviated light and the refracted or deviated light. They go through, they form the two different arms of the interferometer. They recombine in the image plane and interfere. And that interference pattern is what is giving you contrast. So that's what that little thing up there is trying to show. That's probably not so well, actually. The consequence of that, anyway, is that you go from this sort of very low contrast image here to a much more contrasty image in the eyepiece, which you can actually then look to see, in this case, that your cells are coming towards confluence or something like that. So it's, again, it's a sort of interferometer. In this case, it's a collinear one. The important bit with this is that they are aligned. So here in the actual objective itself, you can see it's got this complex lens structure. Somewhere back up here, here's that phase plate. So you want the annulus of the light source to be matched. So here you can sort of see an image showing you the phase plate and the light source, and you can see they're a bit out. So you can use something called a Bertrand lens, which is often just built into the microscope. You just flick it into the light path. And that allows you to look at, rather than the image plane, this back focal plane of the lens, and see these patterns directly. And then on the microscope, there's some little adjusters that allow you to move the condenser around so that it's properly overlaid. And as you bring it into an overlay, you're basically bringing the interferometer into alignment, and you start to see contrast in the sample. Most of the time, we've set that up, and or it's set up by mechanical engineering of the microscope, and you don't really need to adjust it. But once in a while, it does go out. But it is true that you are putting stuff into the microscope, so this does affect slightly the transmission of the microscope, which is more an issue for fluorescence. If you're using a white light source, you just turn up the white light to an arbitrary amount, and you're good to go. Differential interference contrast is a different take on that, and it has the advantage of getting you to finer detail. You don't tend to see this sort of haloing effect around objects. So it again works. You've got a white light source down here. You put it through a polarizer, so that gets you a linearly polarized light. That goes through something called a Wollaston prism, or a Normarsky prism. They're known as in sort of their variation of that particular type of prism in microscopes. And that prism is basically at 45 degrees to the polarizer. And that allows you to split the light into two different polarizations, basically left and right, horizontal, vertical, whatever you want to call it, two different polarizations. And the other thing that this Wollaston does, it shears the beam. So the left polarization and the sort of right, or the vertical and horizontal, travel through the microscope at very slightly different light paths, displaced sideways. And that displacement is of the order of a couple of 100 nanometers. It's very small, but that allows you to sense differential phase objects on the microscope stage itself. The light goes through the object, collected by the objective. It goes back through another matched Wollaston or a matched Normarsky prism, which recombines the light, and then you look at it with an analyzer. The analyzer is crossed with this polarizer down here. So in normal circumstances, if there was nothing going on the specimen, that one will be crossed with that one, and you'd see nothing through the eyepieces. If the specimen is deviating these two beams slightly differently, then the interferometry comes into play, and you get constructive interference and what that be. What it looks like in practice is something like this. So you've got to imagine these two sheared beams. If they're going through the same thing, they effectively see the same light path, and there's no relative phase retardation. If they go through the edges of objects, you can see that one beam could go straight through, and the other one is slightly retarded by going through the edge of the cell. If it's going through the body of the cell, then they're both the same again. So it's this relative retardation of these two sheared beams at the edges of objects where all the magic happens. And then this is what the images look like. You see this sort of funny pseudo 3D type effect in your eye, but you generate a lot of contrast. And you can see that sort of up here, the contrast on this edge is light, and the contrast on this edge is dark. You can tweak the microscope around the different biases in the way the phase interferes, so you can change the sort of how it looks in the eyepieces. But what is remarkable is the amount of detail that interferometer can give you. So here's a sort of slightly small, a small variation in depth in focal plane, and you can see there's lots and lots of contrast that this sort of microscopy can generate. Okay, so moving on. we've looked at how you get detail, how you get fine detail out of a microscope. Now we're going into fluorescence, which is really adding specificity. How do you know what it is you're looking at? Are you looking at cytoskeleton, or nucleus, or mitochondria, or whatever? Fluorescence is a way of adding that extra bit of knowledge, knowing what it is you're actually looking at. So we can basically target particular proteins, molecules of interest. We can target cellular compartments, all sorts of things. And we know by that targeting what it is in the image. So typically we can use antibodies, some natural occurring toxins, binding partners, green fluorescent proteins and whatnot to highlight those bits and pieces in the sample. And it can be used at all scales. I mean, what's remarkable even here is, the era of microscopy without fluorescent proteins, without genetic encoding, is not that long ago. But that's really changed the way we can do microscopy, the advent of those sorts of proteins, fluorescent proteins. The advantage of fluorescence, well, we get good resolution, but we haven't lost it, and there's no compromise on resolution. High sensitivity, so modern microscopes with a camera or whatever, you can detect the fluorescence from a single fluorescent molecule. Very high specificity. We're putting in a protein, a GFP protein construct. It's only that protein that's got the GFP on it. And most importantly, it's compatible with living things. So this is the thing that distinguishes light microscopy from fluorescence microscopy from electron microscopy. No matter what electron, the advantages of EM isn't always going to kill your sample. Light microscopy, we can carry on working for days at a time if we want to, which is what the circadian rhythm people are doing effectively. How does it work? We've got some fluorescent object, either a small molecule dye like Alexa 488, which is the structure there. That's somewhat below a nanometer in diameter. Green fluorescent protein. That barrel is sort of 4 nanometers across, of the order of that sort of size. And attach those to our target of interest in the cell. These dyes sort of have a excitation spectrum and then an emission spectrum. So we can come in with sort of blue light here and generate green light here in this example. And they're typically sort of these sort of broad spectrum here. What's interesting about that is that immediately leads to some other thoughts. So we can think about the way that those dyes work in terms of these Jablonski diagrams. So we've got the ground state, you put in some energy in the form of a photon, you get up into the singlet state. And it's this sort of substructure here that leads to the broadness of the emission spectrum. But we get down from the singlet state with the emission of a photon, but you can sort of see it's coming from a lower energy level than the one we went. But it also is useful because it sort of allows us to think about other things that go on, like photo bleaching. We can get intersystem crossing into triplet states. We can just get returned to the ground state without emission of a photon, so the sample just heats up effectively, things like that. All the engineering of dyes is all about limiting all these other parasitic processes so that just the emission is the one that happens most of all. In particular, we sort of want to try and get rid of photo bleaching. But yeah, as I said, you've got these sort of emission spectra. But because we've got quite a lot of space in the visible spectrum, we can add multiple dyes. So here we can start to look at molecule A behavior in a sample relative to molecule B and molecule C, distinguishing them by their emission colors. So this is a very, very useful property of a fluorescent microscope. The fact that we can look at multiple targets at once. How it's actually done in practice. is using a sort of filter set. So we've got a light source back here, which these days is usually an LED rather than a halogen, rather than an arc lamp, mercury arc. Comes into the microscope and it reflects off a mirror that reflects one color light and transmits a different color light. So we've got, say, blue light coming in here, reflects off that object down through the objective onto the sample. The sample emits fluorescence, comes back up to that mirror, dichroic mirror, and goes straight through it because it's a different color. So blue light reflects, green light goes straight through. We've got a filter up here to clean up the emission spectrum, and then we look at that through the eyepieces or onto a camera. So those filters are all sort of in this little block of plastic or metal inside the microscope, and we can switch between them to look at different colors. So they sort of typically look like this. So there's a dichroic filter on an angle, excitation filter, emission filter, and then you can select a bunch of those inside the microscope to see the different colors. So the lead on from fluorescence was actually confocal microscope, which is how to get clearer images. So there is a problem in biological samples, which is to do with scatter. When we look into objects, the light doesn't just travel in a straight line. A lot of it just gets scattered around. In fluorescence, the excitation light is also hitting all the other fluorescent molecules above and below the focal plane, and they're all starting to emit fluorescence. So we're flooding a sample with blue light, and we've got this ideal focal plane where we want to look at everything, but above and below that focal plane, there's generation of fluorescence from all the molecules that are sitting there above and below them, and those are creating a sort of veiling glare that swamps what we're trying to look at. So that's sort of illustrated in this cartoon. So here's the microscope objective. We've got this small area, the field of view of the microscope down here, which is the stuff we're actually interested in seeing. But above it and below it, we're generating this huge amount of fluorescence. And that sort of obscures the detail from in here. So that is that's the property of all fluorescent microscopes. In reality, if you've just got a single monolayer of cells, it's not too bad. You'll pretty much be able to see what you want to see. But once you go into a thicker tissue and stuff like that, this really does become a big problem. Now, we'll step back away from this. What was the sort of genesis of the confocal microscope? How does it deal with this problem? So there's this chap Minsky in the sort of 50s. He wanted to look at the detailed structure of the brain. Now, he was interested in effectively what we talk about now as AI. How does the brain work? How do we make neural networks? Things like that. So he thought that if he could see the detailed structure of the brain, he could get an insight into how we compute things in our minds. So he wasn't particularly a microscopist per se. He was much more interested in this object, this problem. How does the brain work? But he thought about this whole problem because basically in brain slices, out of focus fluorescence was obscuring all the detail that he wanted to find. So his reason was, why not illuminate the sample point by point rather than imaging an entire field of view? That would deal with part of the problem. But his big insight was to realize that if he put a pinhole into his system, he could also get rid of the out of focus fluorescence and scatter in one fell swoop. And with that reasoning, the confocal microscope had been invented. So it's not just the fact that he's gone to point-like illumination, but he's also put a pinhole in his device. So the general principle is this. You've got a light source, you get a collimated ray of light, comes down to the objective, is focused to a point. Somewhere in the optical train, you've got a scanning mechanism, so that allows you to do a raster scan of the object. it's a single point illuminator, so it's not something you can look at with the eyeball, but you can put a detector and then create an image via electronic means. But up on the detector plane, he's matched this focal plane to a pinhole in the image plane. So only light that comes from the focal plane gets through the pinhole and onto the detector. Any light that's coming from above or below will be out of focus when it gets to the pinhole and be blocked by the pinhole. So the pinhole, even though it's a sort of two-dimensional object, is actually a mask in 3D space. It only lets stuff through from that point there. And then the name confocal comes from this idea of conjugate plane. So it's the same focus. So it's effectively the sample plane and its image plane. But again, it's 1 point illuminated, 1 point detected. So you have to synthesize the image via electronic means. That's what his microscope actually looked like. And it's a right contraption. Most of it is this whole gantry here, that's his scanner. So he's got basically the sample was on these little prongs here. And he got two objectives. You can see there's the sketch of it from his patent. He's created a point source of light up here. Illuminates a point source in his sample. It has to go through a pinhole before it gets to the detector. And then this is his scanner. So this electromagnet makes it move in that direction, and that electromagnet makes it move in that direction. And with that, he built a confocal microscope. It didn't solve the problem of how the human brain works, but it's been an extremely useful tool in cell biology and biological microscopy in general. So this is a problem it's solving. Here, you got some object which is obscured by all of this out of focus fluorescence. You stick the pinhole into the system, scan, and suddenly you can see all the detail. The reason it works so well is because of this two factors, really. The intensity of illumination as you go out. So if we go from this point here to this point here, if we consider the intensity in this plane, this patch of light is getting bigger as scales with z squared. So the background goes down very quickly. Likewise, up on the detector side, the detection efficiency falls off with z squared. So in combination, you've got this z to the 4 factor, which is why it's so good at rejecting out of focus fluorescence. So it's a massive increase in contrast due to these two effects. So illumination intensity and detection efficiency contribute to the idea of confocal sectioning. And that's real property of this object, of this microscope. You get a depth resolved slice out of a 3-dimensional object. If you take a stack of depth resolved slices and put them back together again, then you can make a 3-dimensional rendering of your object. And it's been an extremely useful tool. These microscopes languished for a long time without sort of making much impact. But at the LMB, John White, Brad Amos, Mick Fordham, and Richard Durbin decided to build one. This one's, here we go, 1986. That's what it looked like. To make head or tail of it, here's the microscope itself. The specimen is on here. And there's the microscope, very simple microscope. All of this lot back here is the scanner. That's the laser light source that they used. And then the detector is back here. And this thing here is the pinhole. But to show that in schematic, this is what they had. They got a beam of light, collimated beam of light, got sort of an X mirror. Well, this is a Y mirror, slow scan direction, imaged onto another mirror that scans in the sort of X direction. It goes through the telescope into the microscope and thence to the sample down here, the fluorescence comes back through this whole mechanism. Because it's gone back through the scanners, the fluorescence, by the time it comes back up here, is now a stationary beam, just like the beam going in was stationary. It goes through a pinhole and onto a detector. And that design was eventually commercialized by Bio-Rad. That's a picture of the sort of MRC 600 confocal microscope, this thing here. So all the scan optics and detectors are in this box here, and there's a laser sort of under the table. And it was an add-on you could just sort of plug on top of the eyepiece of a regular microscope and get a confocal image. In those days, the computers were staggeringly expensive and could hold one image. That's something called a frame store, so they're pretty crude by comparison to today's objects. But they're great for working in the lab because we can look at multiple color dyes with very fine, with high resolution in and resolve in 3 dimensions, and then we can render those in 3D as well. So you end up with sort of images of stuff like that. So you can take, this is built up from a series of confocal stacks and then just rendered into 3D. So in the confocal microscope, we are back to point sources, imaged as point sources. So now we're really seeing the diffraction effect. So we have a point-like object. Its image is a diffraction pattern, which in sort of where height is intensity, it's something like this. So this is known as the Airy pattern, colloquially, and or otherwise known as a point spread function. And this is the thing that has extent. So we can have a very small object, but the image of the object is a few hundred nanometers across, as determined by Abbe's formula. In the confocal, we're doing various things. So we're focusing light down to a spot, so we can't get an infinitely small spot. We've got this point spread function. That then is scanned around, and that generates fluorescence. The fluorescence goes through, and we put a magnified version of it. So we don't have 500 nanometer pinholes. We have a sort of finite pinhole in the microscope. And that the size of that pinhole is effectively determined by the magnification of the objective, which might be 60x, 100x. The magnification of all the optics in the scan, in the confocal scanner. And that ends up being a much more usable number, like 50 to 100 microns in the confocal microscope. So this is something that we can feasibly adjust to match the properties of the light we're using. All of those things are sort of instrument dependent and somewhat variable. We can sort of take away all of that and just work in this thing called area unit. So the area unit is effectively where this first minimum of that diffraction pattern is. So if your pinhole is matched to that diameter from there to there, that's called 1 area. So now it doesn't sort of matter if you've got confocal from manufacturer A or confocal from manufacturer B or whatever. 1 area is 1 area. So that just allows you to know how big your pinhole is relative to the point spread function of the microscope. And typically for a confocal microscope, you set the pinhole at 1 area. Conceptually, you can sort of think of it like this. You've got this diffraction pattern and a pinhole. If the pinhole matches that central maximum, that's 1 area. If you open up the pinhole, that pattern doesn't change. You've just got a bigger hole. Most of the time, it doesn't make too much problem. But what's actually happening here is that bigger hole lets much more of the background fluorescence through. So this is obscuring the detail you want to see. as you open it up, you're losing that confocal effect. You're also losing the ability to take out a depth resolved slice from a three-dimensional object as well. There's a sort of trade-off for this. As you shut that pinhole down, you go from one area and shut it down, you're going to lose light throughput, but you will gain some resolution. So this sort of one area is the happy medium. So there's usually in the confocal software, there's a one area button. You just hit that, and that gives you sort of the best balance between collecting light and optical sectioning and resolution. Okay. The other thing that scanners have is this ability to zoom. So you can see, if you want to get high detail, you need a high NA lens, and then with that lens, you'll see a certain field of view, and then you can change how the scanners in that in the confocal unit access the field of view of the lens. So you can sort of draw a box that's equivalent to a large part of the field of view. You might get 512 by 512 pixel array, or you could draw a little box and zoom into it. So that's, you can basically zoom in with the software and the scanners. On the screen, the image looks exactly the same size. It's blown up, to whatever your screen size is. So you just see, the same array size with finer detail in it, effectively. But what we're getting into is sampling theory. Have you actually collected the finest detail that that object, that confocal microscope will give you with 512 by 512 pixels? And you can think about this problem like this. We've got a sort of focal spot, the PSF, it's got a certain diameter in X and Y, and in Z it's bigger, so it's sort of rugby ball shaped. Sampling theory tells us that, well, let's actually go back one. So Abbe's resolution criteria told us that the resolution was 1.22 lambda over 2NA, which is basically half the diameter of that spot. Sampling theory says that we need 2 pixels to sample the finest resolvable detail, minimal resolvable object. So we need 4 pixels across a PSF like that. So that is the condition in terms of sampling and stepping our image, our spot to where we can actually capture the detail that the confocal will give us. And that holds true in both X and Y and in Z. Effectively, we need to do that sort of sampling density. You don't necessarily know this when you're operating the microscope, but luckily they give us a sort of button to press that gets you to this situation without you having to calculate all the numbers. And typically, you're going to see in a confocal microscope that if you're capturing the diffraction limited detail, you need pixels at around about 100 or so nanometers. I mean, often it can be smaller than that. What's actually going on in the sample is that PSF is sort of slowly stepped sideways by units of the pixel spacing, and in Z, similarly, with a lot of overlap. In the image, you just get a value in a grid. So you have no sense of the size of the PSF relative to the grid that the image is displayed on in that pixelated image. And that's the difficulty here, because you don't know what's going on with the scanners. So you just have to be aware of that. Make sure you're sampling properly with Nyquist. So the thing that goes wrong is this. If your step sizes are too large, if your interval from one pixel to the next is too large, you never image that bit in the middle. You never image these gaps. So you don't have you just haven't captured detail. Right, we're reaching the end of my slot. I'm going to skip this bit. There's a lot of things you have to get right in confocals, and actually that's what goes on in training. It's just about how to set up the confocal so you correctly capture the in the electronics a proper representation of the object that's fluorescing on the microscope stage, and there's quite a few views of variables, which is really just what I want you to remember: how much light you put on, how much fluorescence comes out of the sample due to labeling, the amplification in the detector, and then digitization. And you have to get all of that right to get a decent image. So I'm going to skip that for the time being and just go to this point here. You've got a certain dynamic range in your sort of electronics, which is governed by the digitizing circuitry. If you go out of that range, you've lost the detail. So if I'm too bright, if I saturate the system, I cannot digitize what's going on up here. Likewise, if I don't set the 0 level correctly, I've lost the ability to capture this information. So these two things have to be right. What that ends up with is clipping rather than sort of seeing the natural variations near the noise floor. It's just clamped at 0. The bright stuff is just saturated. These are all 255. So you've got to make sure you adjust the microscope to correctly capture the dynamic range of the object within the system. So there are tools that help you do that. And that's shown to you in training as well. Right. I've got a last section to do, which is quite quick. If you want to leave, you're free to leave. I'm just going to go on to multiphoton. So multiphoton microscopy is all about solving another problem, which is light scatter. So no matter how good the microscope is, if the sample itself just randomly deviates rays, you can't get a good image. And that's a problem when you're trying to go into thick samples, so kind of imaging tissue. But this problem was addressed by Denk and Strickler and Watt Webb with this 2-photon laser scanning fluorescence microscopy. And this was a way to really try and deal with the deleterious effect of scattering. So you can see light going through your hand if you stick a torch on it or something like that, but you just can't image any detail. So how do you get rid of that problem? And this is what's going on. Rather than using one photon to excite fluorescence, so this is a typical confocal microscope type scenario. You can see the light coming down to the focal plane and then going out of focus afterwards. Here you see this big cone of out of focus fluorescence, but also there would be in tissue, there's a big haze of scatter as well. In the 2-photon microscope, you require 2 photons of much longer wavelength to simultaneously arrive at a fluorescent molecule in order for it to fluoresce. And that has a very low probability of happening unless the intensity of light is extremely bright. So that means it only happens at the focal point. The other thing that they do with the light is rather than bathing it continuously, is they use pulsed light. So what's going on from a sort of Jablonski diagram is something like this. You're in the ground state. You have 2 photons effectively simultaneously arriving of much longer wavelength, lower energy, which are sufficient to get the molecule up into the excited state, and then it can emit. So this process has a very, very low probability. So what you need are basically very high peak power and low average power lasers. So you concentrate all the photons into a little period of time, but the average power is low enough that you don't fry the sample. And then you focus it down to a small spot, and that combination means that fluorescence is only excited at the focal spot itself. That frees you from some quite interesting constraints. Effectively, the scan mechanism tells you where you are in space, and then all you have to do is just collect light from that single spot, and that's the information you use to get an image. The difference is that that light there is not confounded by a haze of scatter, because obviously if you were including this stuff and this stuff, you would be effectively smearing out the image again. But because you know that the light is coming from the focal plane, from a single spot in the focal plane where you know where it is in three-dimensional space, you can get a scatter-free image. These high peak power, low average power spots have typically come from a mode-locked laser where you get 100 femtosecond duration pulses, 80 million pulses a second with peak powers of staggering numbers, 100,000 watts, but they only last for 100 femtoseconds. And you combine that with focusing, so concentrate in time and space, and you get 2 photon excitation. So that really deals with this problem of scatter in tissue. So now you can start to do good imaging inside intact tissue. And the reason it works is because near infrared light, these lower energy photons don't have anywhere near the same amount of scattering. So there are some advantages of 2 photon. You still get this section effect because it's only at the focus. You don't need a pinhole because you're not exciting out of plane. You're coming in with low energy photons in the near infrared, and looking at visible emission in the green, blue, whatever, red. So there's lots of separation. You don't excite stuff out of plane. And it also happens that you can use a single wavelength for many different dyes. And then finally, near infrared light has deeper penetration into tissue. So you can get into up to 1 millimeter in brain actually these days. And then the scattering business, absorption rather. There should be a scattering rather. Okay, well, in tissue, the problem we have is water absorption and things like hemoglobin. And then in this window, in the near infrared, between sort of 700 to 900 or so, that absorption is low. So this microscope works well in that part of the spectrum where the normal contrast agents of blood and water do not absorb. We're exciting at usually sort of 850, collecting 500. There's massive separation there, so we can easily, that's very convenient for emission filters. We can just have a general broadband filter and collect anything we like. Here's an example. So this is a bit of mouse aorta that has atherosclerotic plaques on it, which have been stained with, I think that's Nile red on these things. So normally you'd have to sort of open the thing up and then take an image and then try and measure the area of these plaques or something like that. In multiphoton microscopy, you can actually image all the way through the wall of the aorta, and you can see the elastin structure in green, which is autofluorescent. Collagen shows up as second harmonic generation on the outside. And you can basically, with a 2-photon microscope, image the entire structure of the aorta. But it's quite nice. I hope this one works because what it shows you is what the elastin structure in the aorta looks like, which you really don't get from traditional microscopy. So this is an advantage of a 3-dimensional microscopy. So now you can start to understand how immune cells, for example, can get from the outside of a blood vessel to the inside, because there are actually windows in there. This whole business of multiphoton led to another field, which is generally called intravital microscopy. So how you can actually look into live animal models. So in the mouse, people have used multiphoton microscopy on sort of all of these target areas, looking to lymph nodes, the kidney, skin, eyes, lung, brain, all of those things. And you can look at those living samples. And that's been particularly powerful in the field of neurobiology, where multiphoton looking into brain tissue has really generated huge amounts of insight into how circuits in the brain work. And the ability to work on freely behaving mice has been a massive game changer in that particular field. For that, you do need to make a window into the brain. So take a small hole or remove a bit of the skull, but then you can actually image into the brain of a mouse. And you can see imaging down the layered structure of neurons. Now, obviously, there's a lot of ethical concerns about this sort of work. So that's all done by other in parallel with all of this. Let me just say that, and I just want to look at the technology, what it allows you to look at, so you can actually see neurons firing in the brain. This one is looking at the immune system inside a lymph node in the thigh of a rat. There's a lymph node here, so this is a preparation where you can actually see what's going on in that lymph node. This was a stunning, it was just stunning that somebody could do this, because what they did was they took the immune cells, B cells and T cells, and labeled them and then put them back into that rat, and then watched them behaving. I can only really show you 2 channels here, but what you're actually seeing is activation of the immune system. Oh man. Okay. Oh, that's a shame. Well, anyway, you can see B cells and T cells contacting each other and stimulating the immune response in real time inside a live animal. You can get into the lungs if you're prepared to do fiddly surgery. You can externalize the kidney and look at what's going on in the kidney. So this was a very interesting experiment because it resolved a discussion about where albumin was filtered in the kidney. This is actually my last slide, so I kept you long enough. I hope this one works. Okay, that's it. I've kept you long enough. Thank you very much for your attention.